Hello and welcome. In this video I'm going to go through the required practicals for Biology Paper 2. There are just 2 for combined science and 2 extra ones if you're doing separate science. Reaction time required practical. As reaction time is so quick, it's very difficult to measure with a stopwatch. So instead we're going to measure reaction time using the ruler drop test. For example, we could ask the question, how does caffeine affect reaction time? Now this is just one example of a factor that we could investigate. You could well in the exam be asked about something like distractions or tiredness or something like that. So this here could change and become a different factor. So let's go through the method. First of all, you'd work in pairs for this practical. Person A will be the person who is dropping the ruler and person B will be the person who is catching the ruler. Person B should sit down and rest their dominant hand on the table with their thumb and fingers hanging over the table. Person A should hold the ruler vertically with the base of the ruler in between person B's thumb and forefinger. This is an important control variable because you need to make sure that you drop the ruler from the same height every single time. Person A will drop the ruler without indicating to person B when this will be, and person B should catch the ruler as quickly as possible. If the ruler falls through just a short distance, this would indicate a fast reaction time, and if the ruler fell through a longer distance, that would indicate a slow reaction time. We can then use a conversion table to convert the distance the ruler fell through the hands into a reaction time. So this will turn the centimetres from the ruler into a time in seconds. We would then repeat this 3 times and calculate a mean reaction time. So you've got the reaction time for person B. We're then going to change this factor here, the amount of caffeine they've drunk, and then we're going to repeat the experiment. So the next step, person B should then drink a set volume. It doesn't matter how much you put, but I've just suggested here 200 ml of caffeinated Coke. They should wait 5 minutes. Again, you should just suggest a sensible amount of time here, and then repeat the ruler drop test 3 times and calculate a mean reaction time. Repeat the investigation using 10 other people, so you could swap around so that person A and B swap around, and then find other people to repeat the investigation as organism distribution required practical. There are 2 different ways that we are going to look at to sample organisms. The first one is random sampling and the second one is transects. So random sampling might be used to estimate a particular population size. So for example, you might get a question a little bit like this that says, describe a method to estimate the population size of daisies growing in a 200 metre by 100 metre school field. These numbers here will come up a little bit later on in our calculation. So we can't count every single daisy growing in the school field. It would just take too long. So we need to have a method of sampling that is completely random, and if they talk about why in the exam, it is to avoid bias. So as we said, we can't count the number of daisies in the whole 200 metre by 100 metre field, so instead we measure out an area of the field using a tape measure. So here I've got a 5 metre by 5 metre area, for example. We then divide this grid up into squares using string and number those squares 1 to 100. So our grid might look something like this. Then the most important thing with random sampling is we use a grid called a quadrat. Now the one I'm going to use in this example is a 0.5 metre by 0.5 metre quadrat. In the exam it may well be a different size, but that's absolutely fine. And we need to choose where to place our quadrat within this grid. So again, it would take too long to do all 100 squares and count the number of daisies in each square. So what we use is a random number generator to select a square, and you can get those on calculators or phones, and it will pick a number at random for you. So we place a quadrat on that selected square and count the number of daisies present, and then we repeat the steps for perhaps 9 more random squares. So we might do 10 in total. So let's say our random number generator picked us out the number 9. What we do is we take our quadrat and we place that on square 9 and we count the number of daisies present. We would then repeat that for another square. For example, if our random number generator there picked us up 47, we'd place our quadrat on 47, count the number of daisies present, and again, for example, 72, again counting the number of daisies present. Now we'd repeat that for around 10 squares. So we've got different quadrats here, and we've counted the number of daisies present in the squares. And we're going to then calculate a mean number of daisies per quadrat. And because we're going to use this number again in a minute in a calculation, I'm just going to leave it as a decimal rather than rounding it as I would do normally with a mean. So we've got a mean number of daisies per quadrat, and now we need to use this to estimate the number of daisies in the whole school field. So we need to know our area of our quadrat, so we do 0.5 times 0.5, and that's 0.25 metres squared. We need to know the area of the school field, so given in the question it was 200 metres by 100 metres, so 20,000 metres squared. The next thing that you need to work out is the number of quadrats that would fit in the field. So we've got the area of our field divided by the area of one quadrat, and that gives us 80,000 quadrats. So if 80,000 quadrats fitted in our field, and the number of daisies in each quadrat was 0.8 as an average, then the estimated number of daisies in the whole field would be our number per quadrat multiplied by the number of quadrats that would fit in the field. So 0.8 times by 80,000 would give us a total estimate of 64,000 daisies. A couple of key points then. Increasing the number of squares sampled will increase the reliability of the population size estimate. They may use the word reliability or they may use the word accuracy, but the more quadrats that you put down on that field, the more reliable your estimate is going to be. Now, for some organisms, calculating percentage cover might be more appropriate than counting the number of organisms. For example, you may well have seen something like lichen growing on trees or walls. If you wanted to sample lichen, you might use a really small quadrat, so just perhaps a 10 by 10 centimetre grid. In the previous example with daisies, we would look at one random square which we'd selected by our random number generator, and we calculate the number of organisms inside. However, with lichen, we can't do this because you can't see individual organisms. So instead, you might need to count the number of squares covered by the lichen. Now, all of the whole squares you can just count up like so, and then we will look at any partial squares that we've got, like perhaps this one and this one, where they could fit together roughly to make a complete square. And we would continue to do this, estimating the total number of squares that are covered by the lichen. So when we've got this, we use the equation the number of squares covered divided by the total number of squares times 100, and this will give us our percentage cover. So if I've estimated that 30 squares in total are covered by the lichen, there are 100 squares on the grid, so 30 divided by 100 times 100 will give us an answer of 30% coverage. Another way we could sample the distribution of organisms is using a transect. So for this we might be asked questions such as, how does light intensity affect organism distribution? Or, how does the distribution of organisms change as you get further from the shore? So let's take this example in terms of seeing how light intensity affects organism distribution. So under the tree, the organisms will be subject to more shade than further away from the tree. So we can sample the organisms at regular intervals and see how the light intensity affects their distribution. So what we do is we'd lay a transect line, this could be a tape measure or some string, along the ground starting at the base of the tree. We would place a quadrat at the 0 meters mark and count the number of organisms, so for example daisies, or it could be something else that you're looking at, present in the quadrat. We would use a light meter to take a recording of the light intensity and record this in a table. We then repeat steps 2 and 3 at regular intervals along the transect line, for example every 2 meters. So we place our quadrat and our light meter here, 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 each time counting the number of organisms, for example daisies or whatever we were looking at, and calculating the light intensity. Now I want you to imagine that we're sampling in a woodland, and along the transect away from the tree there's lots of long plants and long grasses. Now in this case we simply can't place a quadrat on top of the long grass, okay, because the grass is just far too long. So you might see in the exam them talking about counting the plants that are just touching the transect line. So if you've used string and pulled that along the long grasses, you might be simply counting the different species that are touching the transect line. Another thing to look out for, don't be put off if they talk about wanting to measure something vertically. So for example they could look at how height affects leaf size or how height affects lichen coverage. In this case, think about doing the practical in exactly the same way. So you may use a very small quadrat and measure percentage cover of something as you go up, or you might simply, if you're measuring leaf size for example, see any leaves that were touching the line and measure their size as you go up and see how that changes as you go up the transect. So don't be put off if they're talking about one vertically going up a tree or up a wall or something like that. It's exactly the same process as the horizontal transect that we looked at before. Plant growth require practical, and this is for separate scientists only. When you're looking at plant growth in a lab, you're going to want to use a seed such as mustard seeds or cress seeds that are going to grow very quickly. There are a number of different factors that you could investigate to see the effect that they have on plant growth. I'm going to go through a few of those, but you have to be resilient in the exam because they could talk about a number of different factors. The first one we'll look at is this question, which says, how does light intensity affect plant growth? So for this practical, we would set up a Petri dish, and inside that Petri dish you could have a small layer of soil, or you could simply just have some damp cotton wool. Seeds will grow very easily, so cotton wool would be absolutely fine. And you would put a certain number of seeds on there, making sure you keep that constant. So I've put 5 seeds on the dish, and if we're looking at light intensity, we could change the distance between the lamp and the Petri dish, for example. So our independent variable would be the distance between the light and the Petri dish, and this would therefore change the light intensity reaching the plant. Our dependent variable would be the height of the plant, and control variables would include the same number of seeds per Petri dish, the power of the lamp, the type of seed that we're using, the volume of water that we add daily, and the temperature. And depending on the practical, we might want to measure the rate of growth, and we may want to measure the height of the plant every day, or we may just want to leave it for a set number of days and measure the height of the plants afterwards. So to measure the height of the plant, you'd put your ruler so it's touching the cotton wool or the soil or whatever you're using, and hold the plant gently. So you can see at the moment the one that the ruler's next to is slightly bent, so you'd hold it gently up against the ruler to get an accurate height. So you'd measure the height of each of the seedlings in each of the discs, and you'd have an average height that the plants have grown under each light intensity, which you would then record in a table. A different question that you might want to ask might be, how does the direction of light affect plant growth? So this time keeping the distance between the lamp and the Petri dish the same, but changing the direction. So perhaps having the light coming in from the left or the right, or from directly above, for example. So in this case, our independent variable would be our direction of the light source. Our dependent variable might be recording the direction of the plant growth. Our control variables, important things as before, like the same number of seeds per Petri dish, the power of the lamp, the type of seed, the volume of water added, temperature, and in this case the addition of an extra one, which is the distance between the lamp and the seeds. And then because of phototropism and the response to light, we would have the plant growing and changing direction towards the light source. And in the exam you may well be asked to draw drawings of the seeds to show how you might predict them to grow or as a result of a particular experiment, and in which case you would use clear pencil drawings. Again, linking into tropisms, this case geotropism, we might want to look at how the angle of the Petri dish affects plant growth. And in this case we might want to set up the Petri dishes at different angles and then record plant growth. In this case our independent variable would be our angle of our Petri dish, so we might want to do 180 degrees, 90 degrees, 270 degrees, etc. Our direction of our plant growth would be what we are going to record down after our experiment, and our control variables would be, again, similar things to before, and make sure we're keeping the direction of the light source the same, and also light intensity or background lighting we'd want to keep the same in this one as well. As a result of this experiment, then we may expect results a little bit like this, whereby the shoots are responding to gravity and growing upwards in all cases. Now you may well in the exam also see experiments where you can observe the direction of root growth as well, so look out for those ones too. Decay required practical for separate scientists only. In this investigation we're going to look at setting up an experiment to investigate how temperature affects the rate of decay. Now when we're looking at calculating rate, we're going to use an equation right at the end that says rate equals 1000 divided by time, because we're just looking at a colour change and this is the equation that we need to use. So for this experiment we're going to set up a few things first of all. We're going to set up 3 water baths, and I've just suggested these temperatures. It doesn't matter which ones that you suggest. So we've got one of 20, 40 and 60 degrees. If you wanted to have a cooler temperature as well, you may well set up some water in a fridge or some water with some ice in it to have a look at colder temperatures as well. But I'm going to look at one where we set up 3 water baths, and then for each water bath we need to set up 2 test tubes, or you can use boiling tubes, either is fine. One test tube containing 5 centimetres cubed of lipase, okay, so that will go in here. And the other test tube we're going to put in 5 centimetres cubed of milk, 5 drops of phenolphthalein, which is our indicator, and 7 centimetres of sodium carbonate solution. All of these measurements here we are going to measure out using a pipette, okay, because that can draw up a fixed volume and place it in our tubes for us. Now we're going to then place these into the different water baths. Now the sodium carbonate solution is something that makes the solution first of all an alkali. So with phenolphthalein it will go pink in alkali conditions. That was the closest colour I could get to pink. So we go pink in alkali conditions. And what will happen in this experiment is the lipase is an enzyme that will break down the fats in milk. And when fats are broken down, they are broken down by the lipase into fatty acids and glycerol. And it is this product here, the acids, that causes a colour change. So in the first instance, we have an alkali conditions created by the sodium carbonate, and then later on we're going to add the lipase, and that will break down the fats and produce fatty acids. So this solution here should go from pink to colourless. because that is the change that we have when we're using phenolphthalein. Now in the exam they may give you another indicator, or you may be used to using another indicator, and that's absolutely fine to do as well. So we've placed our 2 test tubes in our water bath here, and what we need to do then is put a thermometer into each test tube and wait for the temperature of each test tube to meet that of the water bath. So we just need to allow it to stabilise and get to the correct temperature. We are then going to remove 1 centimetres cubed of lipase from our lipase test tube using a pipette, and we're going to squirt it into the other test tube containing the milk, the sodium carbonate and the phenolphthalein indicator. Then using a stopwatch we're going to record the time it takes for the mixture to go colourless, and then we are going to repeat this for the other temperatures that we said before. We'd chosen 40 degrees and 60 degrees. And as always is good practice, repeat the experiment 2 more times for each temperature and calculate a mean time to go colourless. Now to change this into a rate, I hinted at the equation earlier, we'll use rate is 1000 divided by time, and that will give us our rate in per seconds, which you sometimes might see as s seconds to the -1. So the independent variable for this practical is temperature. The dependent variable is the rate of decay, and important control variables include the volume of lipase, the volume of milk, and the volume of sodium carbonate. It's unlikely that you'll get the mark in the exam if you simply say the amount of lipase or the amount of milk, so do make sure you're using the word volume when you're talking about your control variables.